All right, this is Concept 1 notes on regulation and feedback mechanisms in our homeostasis extension mini unit. Okay, so just a little overview before we jump in. We talked about homeostasis back in our cells unit when we learned about cell transport and how those are really important strategies that the cell utilizes in order to maintain homeostasis. So, what is homeostasis again? It is the stability of the internal environment and the mechanisms that maintain that stability. So it's keeping and regulating constant and stable internal conditions within the cell at a cellular level, but also just within the organism at an organismal level. So a dynamic equilibrium is maintained via homeostasis. And that means the rate of loss is going to balance out with the rate of gain. So that's kind of a fancy term, but it's really, really simple if you oppose it with static equilibrium. So there's dynamic equilibrium and static. Static equilibrium would be those constant and stable internal conditions are static, meaning they're unchanging. So, for instance, a normal body temperature is 98.6 degrees Fahrenheit. If it's static equilibrium, it's going to stay right on that number. That's what stability is going to be. Whereas dynamic equilibrium more ebbs and flows. You know, your body temperature isn't always 98.6 degrees Fahrenheit exactly. Sometimes it's a little above that and a little below that. There's a range in which we consider a normal body temperature. And it's you're never just constantly at one point. That's what makes it dynamic. It goes above and below whatever our set point of stability is, but it still maintains that equilibrium. So that's kind of the difference there. And something that really affects maintaining dynamic equilibrium and thus maintaining homeostasis is an organism's ability to not only detect stimuli from their environment, but also then respond to those stimuli. And that's something we're going to talk about a lot in this concept. If you remember when we learned about characteristics of life, the ability to respond to a stimuli or to a stimulus in singular form, that is a characteristic of life. And so that's something that all living things do. Homeostasis, like I said, is maintained through regulation at the big organism or organ system level that make up organisms all the way down to that cellular level. And there's a ton of different factors that have to be regulated and maintained. There's your pH, which can range from being on the acidic side to the basic side. There's temperature, which can range from being cold. Remember, cold doesn't really exist. It's a lack of heat, but also to the other end of hot or too high above the stable temperature. There's blood sugar. You can be hypoglycemic, you can be hyperglycemic if you're outside of that normal range. So we're working within parameters here to maintain stability. There's never just one set number. There's usually a range here, and we can be below that normal range or above it. So how do we do this? What are some of these mechanisms we use? Feedback mechanisms have evolved over time, and that is how organisms maintain homeostasis. We look at them kind of as loops. These loops use the output of our system, and then those outputs signal what's going to happen in the input, what's going into that system, so that the response is then either going to stabilize a system or amplify a system. And we classify or categorize these feedback mechanisms as positive or negative. So, positive feedback mechanisms. In a positive feedback mechanism, or you could refer to it as a feedback loop, since it loops around here, the output or product of the system is then going to intensify the response. So it's going to cause the input to then intensify what's happening. Another way is we can say it's amplifying. So a couple of real world examples. In humans, childbirth is a positive feedback loop because what is put out, the product, is then going to intensify or amplify, and we're just going to keep amping it up. So how does that work? Hormones are released that signal contractions, and when contractions are happening, that creates a lot of pressure. That pressure builds and builds and builds, and that signals more hormones. So the output of this pressure is going to cause more hormones to be released. That's going to cause more contractions, which builds more pressure until eventually, of course, the baby is born. An example not in humans, in plants instead, is the ripening of fruit. So when a fruit ripens, this releases a hormone called ethylene. And that ethylene hormone signals surrounding fruit to also ripen. So as those ripen, they start releasing ethylene, which then causes their neighboring fruit to ripen, which releases more ethylene. And it just keeps going and amplifying this system. That is what a positive feedback loop does. On the other side of things, we have a negative feedback loop. In a negative feedback loop, the output or product is then going to cause a counter response so that we can get back to whatever our set point or our normal range is. So we say this is a stabilizing loop or stabilizing mechanism. If we're out of whack, whether it's too high or too low, we're going to bring it back in gear, bring it back to normal instead of amplifying like a positive one does. And there are so many examples of this. Human body temperature, which we also call thermoregulation, water concentration or osmoregulation, blood sugar regulation. And we're going to talk through these in just the next few slides. So one of the things we can kind of use to visualize these feedback loops are actual loop drawings like the ones that I have right here on the slide. So in a feedback loop, we kind of have 4 components. We have your receptor. This is some sort of sensory organ that's taking in and receiving the stimulus from the environment. So of course, then we have our stimulus. Okay, so we have maybe the initial stimulus in the receptor, but also that taking that in can cause another stimulus that's going to evoke some sort of response. We have the effector organ. This is the one that's going to actually do the response. And then, of course, we have the response, which is our effect that's caused by our stimulus. Now, I show the arrows going 2 directions because remember, a feedback loop can be initiated when we're too high outside of our range, so we're above where our range should be, or we can be too low. Either way, we're trying to get back into that normal range to maintain stability. All right, so we're going to kind of walk through a couple of examples. So first, let's look at a thermoregulation feedback loop. So some organisms are ectotherms and some are endotherms. Ectotherms do not have the ability to regulate their temperature internally. Their temperature is the same as their environment. So we see this in a lot of reptiles, whereas humans are endotherms. We have a complex internal system that we're about to talk through that allows us to regulate our temperature despite what's going on in our environment. So even if it's 50 degrees outside, 50 degrees Fahrenheit, your internal temperature can still be 98.6 degrees Fahrenheit. So I'm going to talk you through this feedback loop. Just know that I'm oversimplifying in these examples. I'm trying to make it clear so you can kind of understand what's going on, but there's a lot more going into this than what's pictured. So this is just an oversimplification, but we're doing it for the sake of understanding. Okay, so normal body temperature in a human is considered to be 37 degrees Celsius or 98.6 degrees Fahrenheit. But of course, we can kind of go a little above or below that. No problem. So the hypothalamus in your brain, that is our receptor organ. That's what is receiving signals that say, hey, it's too hot or it's too cold. So let's start with too hot. So when we're too high above this normal body temperature range, we have this stimulus of hot that the receptor, which is your hypothalamus in your brain, receives. Okay, this initiates an action in a couple of different places. We can look at it in your sweat glands. We can also look at it in your capillaries. And what will happen is the action, the effect of the stimulus of hot is you're going to sweat in your sweat glands, which is going to release water, and the evaporation of that water from your skin is what cools you down. Another thing that can happen in our effector organ of the capillaries is that your blood vessels will dilate, and this increases the surface area, which allows more heat to be released. And then that's going to bring us back down to our normal range. Okay, so that's kind of our loop here. Now, let's look at the other side of things. Let's say we're too low. We're below our range here. The stimulus then is cold in the hypothalamus in the brain. And that, again, that can cause a response in an effector, which is your muscles, or also the capillaries. So what could those responses be? Your muscles shiver. Okay, so when your muscles shiver, they're contracting. This requires energy. And anytime we're going to need energy, we're going to have to harvest it, and heat is a byproduct of harvesting energy. So the act of shivering requires energy, and that causes a release of heat as a byproduct, which will then get us back to that normal range. Or our capillaries, those blood vessels can constrict, which will decrease the surface area, which will retain more heat, not let it be released, which will bring us back to our normal range. Okay, so that's just one example in thermoregulation. Let's talk through another, osmoregulation. Okay, so the normal osmolality of human blood is, again, it's a range. It's between 275-ish to 295 milliosmoles per kilogram of solvent. Okay, so what's our receptor? Your hypothalamus, which is then going to actually signal the pituitary gland to release some hormones. So the hypothalamus in your brain, sensory, it's going to signal the pituitary gland. So these are going to be the ones receiving the signal. Okay, so if we're too high, if we're above this range, what does that mean? That means your blood has a higher concentration of solute, aka you're dehydrated. There's less water. So what does that stimulate? That stimulates ADH, the antidiuretic hormone, to be released. So your pituitary gland causes a stimulus of ADH to be released. Okay, what does that ADH do? Well, your kidney nephrons, your nephrons in your kidney, these are the effector organs. They're going to cause an effect or a response. And that response is those nephrons are going to hold on to water. So they're not going to release as much water into your urine. This is when your urine looks really, really yellow. That's because it's super concentrated, because we're trying to retain water so that we can get this high concentration of solute down and we can get you back into your normal range. Okay, well, what if it's too low? If we're below that range, that means you have a lower concentration of solute. So that means you're too dilute. There's more water there. So we're going to release less ADH. So your hypothalamus is going to tell your pituitary gland, hey, don't release as much ADH, that antidiuretic hormone. Then your kidney nephrons do the opposite. They're going to allow water to be released. They're not going to retain water. They're going to allow water to get released into your urine so as to balance it out again and get us back to that normal range. One last example is blood sugar regulation. So your blood sugar, the norm for blood sugar is different depending on when we're talking about. Are we talking about right before you eat, right after you eat, while you're sleeping, that kind of thing. So for the sake of this example, we're going to talk about the blood sugar of a human right before they eat is usually somewhere around 70 to 90 milligrams per deciliter. So what is our receptor organ? It's your pancreas. The pancreas will receive the signal, that stimulus of too high or too low. If it's too high, that means that your blood glucose levels are too high. So the sugar amount that's in your blood is too high. That's the stimulus. That stimulates your effector organ, which is the beta cells in your pancreas. The response then is those beta cells are going to release a hormone called insulin. Insulin causes your body cells to take in glucose from your blood. So when they intake that glucose, it's going to then lower your blood sugar and get it back to that normal range. Now, if your pancreas, that receptor organ, it receives a signal that we're too low outside of our range, that means we have a stimulus of low blood glucose. That is going to cause a response in the effector organ of your alpha cells, which are in your pancreas. Instead of releasing insulin, they're going to release a hormone called glucagon. And what glucagon does is it causes your liver cells to break down glycogen. If you remember, glycogen is a storage molecule for carbs, and 10% of your liver's mass is glycogen. So it's going to tell your liver, break down glycogen, release that glucose from the broken down glycogen into your blood, and thus that's going to raise your blood sugar back again into our normal range and thus maintaining homeostasis. So these feedback loops, what if something goes wrong? Well, if there's a mistake, that's going to throw off homeostasis, and that can have small impacts or can have really great ones. Really, really common example is type 1 diabetes. So in type 1 diabetes, the pancreas produces too little or none at all of insulin. That hormone we talked about back here, which causes your blood cells to take in glucose from your blood and lower your blood sugar when it is too high. So that means we're not getting blood sugar levels down when they're too high. So we're having a regulation issue here. And that's a big problem. And we can go into more of that later. There's actually going to be an activity that y'all are going to do to kind of explore mistakes and what happens when things go wrong. But that's just a pretty common example of a mistake in a feedback loop. Okay, last thing I want to touch on are enzymes. Enzymes are so important for regulation and maintaining homeostasis. So what are they again? Maybe you remember, maybe you don't. Remember, enzymes are proteins that act as catalysts, meaning they initiate and then they also then regulate chemical reactions. They can make them happen faster by lowering the energy it takes to get the reaction started, which is called the activation energy. Now, that's what they can do, but they can also do the opposite. We can also, by removing an enzyme, we can then inhibit a reaction and thus slow down the rate. So that can happen too. How do they work? Big theme of biology, y'all. Structure dictates function. Enzymes have such specific structures. Remember, enzymes are proteins, and proteins have 4 levels of their structure. They have the primary, secondary, tertiary, and quaternary, meaning they're highly, highly specific. And that insane amount of specificity is going to make them only bind to certain substrates or certain reactants. And that's going to affect how they function and what chemical reactions they then can catalyze. So that specificity is super important. Why are they so important? Well, a lot of reactions are not going to happen at the rate we need them to happen unless an enzyme is present. They pretty much, every metabolic pathway has an enzyme that corresponds to it. And they're so specific, y'all. So here's such a simple diagram of what an enzyme could look like. This shape here is the active site where the substrate that specifically binds to it. It can't bind ones that are a different shape. And we often think of it as a lock and key model. So the specificity of a certain key only fits a certain lock. But really it's more of an induced fit. So it's more like a ball and a catcher's mitt. So when a baseball hits the catcher's mitt, the mitt it not only fits there, but then the mitt will close around it. So it's even more specific than just a key fitting into a lock. But both are great visuals. Okay, so what's their role again in regulation? Well, living systems depend on these biochemical reactions, and all of these reactions occur spontaneously, just at very slow rates. So we need catalysts to speed up the reactions without actually being a reactant or a product. So they're not affected, they're not permanently altered by the reaction, but they play a part in getting it going. No catalyst is going to make a reaction occur that couldn't occur otherwise. It's just going to make it happen faster. And most biological catalysts, like we said, are proteins, meaning they're enzymes. Pretty much every step of a metabolic pathway is catalyzed by a specific enzyme, like the glycolysis, the Krebs cycle, all these parts of cellular respiration. They all have specific enzymes that catalyze each step. So how can we regulate using enzymes? Well, we can regulate metabolism by controlling how much of an enzyme there is, the amount of it. We can turn on and off the synthesis of enzymes in order to then control the rate of reaction. Feedback inhibition is specifically where the output of a process is used as an input to then control the behavior of the overall process itself, usually leading to inhibition. And this is really important because we want our cells to run efficiently. We don't want resources to be wasted. So feedback inhibition and that turning on and off of enzyme synthesis is super critical to maintain that efficiency. We can also regulate the activity of an enzyme. We can also use chemical inhibitors that could bind to an enzyme and then slow down the reaction rate. And then, of course, there's environmental conditions that can play an effect too in an enzyme's ability to function. We talked before about pH and temperature, these environmental changes that can lead to the denaturation of an enzyme, where the active site of an enzyme is changed, and thus it loses that specificity for a certain substrate and can't bind where it's supposed to bind and do then what it's supposed to do. So this, again, super brief overview, but enzymes are incredibly important for maintaining homeostasis. And that is concept 1.